over there. Greetings. It's my great pleasure to introduce one of the most important people here as part of this conference, and that is Dr. Jean-Paul Piernet, who is head of the hospital in Belgium, where he's been working with Phage and Phage for treating a lot of different kinds of things, with Phage from Georgia and with students from Georgia, and also with Bob Blaisdell, who was my student, who had been very much influenced by him. And starting back in 2008, they used Phage in various ways. Phage from Georgia had people from Georgia coming and working with them. We're working back and forth all of the time and are the main reason that things have gotten so far there. So I'm delighted to introduce Jean-Paul Piernet.
So I would like to thank Betty and the organizing committee. And I would like to thank you for having me. So I'm going to talk about phage therapy in Belgium. Okay, the title is a bit weird. Bacteriophages, it's a medicine, Jim, but not as we know it, but I'll explain later why to the younger attendees. I don't think it's working. Okay. So we began thinking about phage therapy in 2003. And as everywhere, to research you need funding. And so we submitted a proposal to the Royal Military Academy to get some money to do research. Back then, proposals, they were evaluated by a bunch of experts, I think 11, I didn't count them. But, and that year, 2003, there were 26 proposals, and they were all evaluated. The color code reflects the scores. Of course, red is always bad, so you should stay out of the red. 11 proposals got funded, the green ones. And where were we? We were down and we scored 8 out of 20. And some of the experts had said had written on the proposal, because it was all written back then, that it was science fiction. Now everything is relative, because the proposal scoring 7 out of 20 was collecting postcards pre-Waterloo. So a therapy from 1920 could be seen as science fiction. So, but since then we obtained funding, and in the 17 years that we are researching phages, we developed an entire chain of phage research, from isolation to therapy over production. And what is special is that it's all under one roof. It's all in the military hospital. But of course, we have a lot of collaborations, for some fundamental research, but we have also a very good collaboration with the Ilyava Institute, and this is very important. So when we started with phage therapy, we didn't want to reinvent phage therapy, so we approached researchers of the institute that was dedicated to phage therapy for almost 1 century. So more specifically, Dr. Zia Kutadeladze, the director of the Liav Institute, and Dr. Nina Chanishvili, the research director. But we had also researchers come over, and they, you can see Dr. Thea Glonti and Dr. Maya Muyabishvili. And Maya is with us, I think, now for 15 years. And they, in fact, coordinate all our phage therapy research. So what we do is everything we do is based on the experience of the Liav Institute. And I always say we just put some BRNS sauce over it. As most of you know, phages are considered to be a medicine, both in the EU and the US. And also important is, and here you can see a quote from the European Commission, what is important is that the existing medicinal product framework is adequate for bacteriophage therapy. So they did not want to create any dedicated framework. So they need to be considered as conventional drugs. And this means that they need to be produced according to GMP. And GMP stands for Good Manufacturing Practice. And after this GMP production, you need to do preclinical tests, and you need to do phase 1, 2, 3 human tests. And then you need to submit a dossier for marketing authorization. There are some companies that are trying to go through this pathway, and they do this with phage cocktails, because they, of course, if you are going to do this entire process, you want to have broad spectrum cocktails that will be applied in several indications all over the world, because this costs millions of euros and this takes 5 to 10 years to come to the market. So you need a lot of time and a lot of money. But the problem is that when a company invests a lot of time, a lot of money, they want to protect these investments. And as you know, phage therapy is in the public domain since the 1920s. And also since the Myriad Genetics case in 2013, we know that it will be very tricky to patent phages. This is the case of those breast cancer genes 1 and 2, and Myriad Genetics obtained a patent, but this was challenged by the Association for Molecular Pathology, and this association, they won, and the judge said that you cannot patent products of nature. There's no invention. So, as I said, still, there are some companies that are trying to go down that pathway. And then after GMP production, they need to do randomized control trials, RCTs. And there have been a handful of these RCTs, and they evaluated defined phage products, and they were also used as standalone therapies. And as you probably know, they showed some disappointing efficacy. And this in contrast with historical phage therapy applications, as we have seen in Georgia and Poland, but also in contrast to recent Western case studies. Now, why is this? So, of course, there will be a positive result publication bias, because people tend only to publish cases with a positive clinical outcome. But what is also special about these cases in comparison with the RCTs is that phages are used as adjunctive therapy most of the time together with Another difference is that selected phages are used, not some generic phage cocktail. So the phages are selected to target the patient's infection. So they are personalized. Another difference is that often they are pre-adapted, and there are even some cases where phages have been engineered to be more effective. So that's why I tend to say that it's not phage therapy that has been evaluated in these RCTs. It's defined phage products. So it is phages as if they were antibiotics, broad spectrum cocktails. So now we come to the title of the presentation. We took this title from a publication Betty and I published last year. So bacteriophages, it's the medicine, Jim, but not as we know it. And this means that, yeah, okay, according to the definition, it is a medicine, because the definition says that any substance showing therapeutic effect is a medicine. But the thing is that these are special antimicrobials, and that the things that make phages special were not used or underappreciated in these RCTs that really tested defined phage products. Now, for the younger participants, so who is Jim? Well, Jim is a fictional character. Jim stands for Captain James T. Kirk of the Starship Enterprise, and on their travels through the universe, they often encountered life, but life that was not as they knew it. Give an example.
Captain, doesn't even look real.
It is not life as we know or understand it. Yet it is obviously alive. It exists.
And it can bear up under full phaser power.
So it is life, but it has some peculiarities, like resisting to phasers, for instance. And have phages. So phages have also some peculiarities, and I will only discuss the ones that we think are relevant for phage therapy. So the first one is that they are very specific. So compared to antibiotics, who usually will target even more species, the phages, they will only target one species or even only a part of a species, and sometimes even a few strains. And this has advantages. So you can choose phages not to target the patient's commensal flora, because a lot of adverse events of antibiotics are due to disturbances in the patient's commensal flora and the intestines, for instance. But the downside is that you need to know which bacterium is infecting the patient. And this can be problematic when you want to use them in emergency setting, where you have, for instance, sepsis, and you need the phages immediately. And I can illustrate the problem of specificity with this slide. This is when you look at phage therapy requests in San Diego and Brussels, and you look at the species that were involved in, I think it's about like 1 year of phage therapy requests, both in San Diego and in Brussels. And you see that in 488 requests in San Diego, 35 species were involved, and in Brussels, in 249 requests, 31 species. So this means that if you have a phage therapy center that wants to cater for these requests, you will need to target more than 30 species. As you also can see, there are some geographical variations, and there will also be variations over time. So let's be very conservative and say that you would need 5 phages to target one species. So this would mean that you would need hundreds of phages in your phage therapy center. And each of these phages would be a medicine that would have to be produced according to GMP and would have been evaluated in preclinicals and randomized controlled trials phase 1, 2, 3. We don't think that this is very realistic to do this. Another example of phage specificity, you all know, or at least the people familiar with phage therapy, the Phage study. And in this study, a cocktail of 12 lytic phages active against Pseudomonas aeruginosa was evaluated in burn wound infections. Now, what is less known is that in an ancillary study, it was shown that 1 in 3 strains isolated from patients before therapy were resistant to the phage cocktail. So we have a cocktail of 12 phages, and already before therapy, 1 in 3 of the strains that you want to target are resistant. Then, of course, during therapy, you also get resistance emergence. So peculiarity 2 is that bacteria and phages are involved in a host-parasite relationship. And it's a very complicated relationship. We have seen this this morning in all these nice talks. So we know that normally phages will not eradicate their hosts. So we need to count on the immune system of the patient or other antimicrobials to eradicate the bacteria. And not to be eradicated, what happens is that phage-resistant bacteria will be selected. And this happens rather fast, faster than against antibiotics. But of course, you know, new phages can emerge to infect these resistant bacteria. So phage co-evolve with bacteria. I want to show you an example of the emergence of the in vivo emergence in the patient of bacterial phage resistance. So this is in a 1-year-old child with Pseudomonas aeruginosa infection, sepsis, after liver transplantation. And during phage treatment, we isolated sequential bacteria. And what you see here is the chromosomes of 6 of them. The green ones are sensitive to the phage. The red ones have become insensitive to the phage. And the phage was phage PNM. And when you look at the red circles, you will see mutations in type 4 pili genes. And type 4 pili are needed for PNM infection. Now, what is also interesting is that often it is said that this resistance will lead to less virulence. But in this case, at least in the Galeria melanella model, we didn't see any reduced virulence. Also interesting in the same patient is that we saw phage antibiotic synergy. So this patient, together with phages, received colistin, gentamicin, and aztreonam. And what you see is the bacterial growth. And so the red line is the bacterial growth without antibiotics, without phages. The yellow line is bacterial growth with antibiotics. So you see there is some reduced bacterial growth. The green line is bacterial growth with phage PNM. And then the blue line is when both antibiotics and phages are added in the same concentrations. So you see a clear synergy. And we see this in a lot of patients and with a lot of different antibiotics. A second example, and this is Pseudomonas aeruginosa nasal decontamination. Now, this is a complicated picture, but this is just to say that in this case, we saw some reduced virulence in Galeria melonella. And we saw this with bacteria that emerge resistance in vitro, but also in vivo, but only when these bacteria are resistant to more than one phage. When you were resistant to one phage, there was no reduction in virulence. What was also interesting is that that our collaborators in the UK, so the group of Angus Buckling, they found that the resistance mechanisms were different. So when using the same phages and the same bacteria, when they emerged resistance in vitro, they saw that this was mainly due to biofilm formation. While the in vivo selected resistant mutants, it was not biofilm formation that made them resistant. So different mechanisms in vitro and in vivo. An example of phage training. So I think about a year ago, we were asked to train phages against the Staph epidermidis isolate. But I will first explain what phage training is. So phages are selected from collections, and then they are matched to the infecting bacterium. This is normal. But then you can go a step further and you can pre-adapt them or train them to be more effective against the patient's bacterium. And there is a study in vitro from Ville-Freeman and colleagues that shows that when you pre-adapt your phages, this will lead to increased pathogen clearance, but also lower resistance evolution. So now back to this request to train a phage against Staph epidermidis, because at that time we didn't have Staph epidermidis phages. We only had Staph aureus phages, and we had phage ISP, which is quite renowned phage from the Eliab Institute. And this phage is known to also target Staph epidermidis, but to a lesser extent. And we saw that this phage ISP had some activity against the patient's Staph epidermidis isolates, but not sufficient for therapy. So then Maya trained the phage to become more effective against the patient's strains. And we call this phage ISP epi. And this resulted in stable lysis for 48 hours without the emergence of resistance at a low MOI. And this adaptation was due to 4 missense mutations. And 2 of them, we have no idea what they do. But what was also interesting is that, so we tested this ISP against a collection of 15 Staph epidermidis isolates, and we saw it was active against 3 of them. But this adapted phage was only active against the patient's strain. So it got very specific, specialized to the patient's strain. But this was a personalized phage. So now we come to the pathway that we would like to follow in the hospital, and this is the personalized pathway. And you see on the left collection, a bank of phages. See on the right a patient.
Apple Cash is hier helaas niet beschikbaar.
Okay. And then, so a sample containing the infecting bacterium of the patient would be sent to a lab where it is screened against the phage bank. Then the phages that show activity are mixed in a carrier, which can be, for instance, in hydrogel for burn wounds, or can be a IV solution, and then are given to the patient. So, and we like this approach because it's, of course, it's more sustainable, and it's in line with evolutionary or Darwinian medicine. It's using the natural controls of bacteria, which control bacteria on Earth, and to use them to control infections. And what we also like is that we think this will also have less risk of persisting or spreading bacterial phage resistance. So to avoid the mistakes we made with antibiotics. Of course, it's a bit difficult to combine this with the current medical practice, because MDs most of the time they want the phages now. So there's not really much time to select, to train. So this is why that we think there is a place for both approaches, to have those broad spectrum cocktails on the market for to be used in first line. And then when you have more time, to go for more personalized preparations. So what is the status today? I think we are now 2 decades into what is called the phage therapy renaissance. And there are still no products, no phage medicines on the EU or US markets. So after 20 years, nothing. And of note is that until the 1990s, the Institut Pasteur in France delivered phage preparations. Of course, you also know that phage therapy is in the media. So there are some famous cases. And this puts, of course, pressure on medical doctors to use them in desperate cases. Because also often patients will find information on the internet about those cases, and they also want phages. And then they approach a doctor, but the doctor has some problems using them because they're not on the market. So, and that's a problem that we try to solve in Belgium. And you see here Dr. Magie de Blok, who was our Minister of Public Health in 2015, and you see her in the Belgian Parliament, where she was confronted with questions from the opposition, the Green Party and the Socialist Party, and they asked her, so how is it with phage therapy? Why isn't it developed? And so there she answered that she was aware of the problems getting phages to the market using the conventional pathway as industrially produced medicine products, but she opened the door to magister preparations with phages, because these preparations, they are not subjected to certain constraints such as marketing authorization or GMP production. And this is also important. She asked her competent authorities to help us set up such a framework. Now, first, what are magister preparations? Well, it's part of European and Belgian law. And the US, you know this as compounding pharmacies, which is the same. And definition is any medicine product prepared in a pharmacy in accordance to a medical prescription for an individual patient. So this sounds very much like a personalized approach. And the active ingredients, in our case, the phages, are called active pharmaceutical ingredients or APIs. And these ingredients must be produced conform the provisions of a monograph. And here you see the monograph that we elaborated with the authorities. It's a short document, and it's a recipe which describes how phage active pharmaceutical ingredients need to be produced and tested. So production and testing is in these 2 pages. I will briefly explain the concept that we have in Belgium. So we have a phage bank, and this phage bank you will find characterized phages, and they are stored using a banking system. Now, to get your phage into the bank, they first need to pass some external quality testing by a Belgian approved laboratory. And this is basically that they need to obtain a genomic passport. So both the phage and its production host will be sequenced, and then the genome will be analyzed by experts. What are they looking for? They are looking for lysogeny-related genes, because you want strictly virulent phages, genes related to antibiotic resistance, or to virulence. They are looking for insertion sequences, transposons, endonucleases. And they also check the host for toxins, etc. They also validate a qPCR that will be used downstream to identify the correct phage, and also to quantify it. And this is the end of the certificate. What is measured is the safety of the phage. And you see the bottom right goedgekeurd. This is Dutch for approved. So when you have this certificate, the phage can go into your bank. Next step, you need to produce the phage as an active pharmaceutical ingredient according to the monograph. And then you need to again send samples to the Belgian approved laboratory for quality testing. And this quality testing is very pragmatic. It's endotoxin content, bioburden, and pH. And then again, it needs to be approved. And now I get this question a lot. So everybody thinks this is GMP. No, they are not GMP. This does not mean that they are not of quality. Yeah? So we produce them in clean rooms. We have standard operating procedures. We have a quality management system. So then once this batch of phage APIs is approved, it can go to hospital pharmacies together with the certificates, and then they can be integrated into magister preparations upon prescription by a physician. And these magister preparations can be just suspensions, mixes of phages, or they can be combined again in hydrogels or IV solutions, or they can be powders, whatever. You see some examples of magister preparations in the military hospital. Next step, of course, was to explain to the pharmacists what phages are, and also to provide them with the necessary information. For instance, on stability of the APIs that we deliver. You can see an example of 3 phages, 14.1, ISP, and PNM, in different solutions that are used for IV injections. You can see that the more you dilute the phage, the less stable it becomes. And then you see also some differences between phages. This is an example of the stability or the activity of 5 phages in 13, I think, yeah, 13 burn wound creams and gels. Because, for instance, the burn wound center, they will often be combined with those creams. And there you can see that in some creams, phages are inactivated in minutes, while in some other creams, there is no reduction in activity even after 24 hours. And you can also see that there are differences between phages. So it's very useful to have this information to know which phage can be combined with which cream. You also have to know, for instance, when you nebulize phages, that not to use jet nebulizers, for instance, because as you can see, you will get a log2 reduction of activity. This means that you will have like 1% of phages active after nebulization, but you have much less problems using mesh nebulizers. It is important to have all this information. You can also dry or freeze dry phages, and this is a project. We are partners in IntelliPhages, and the scientific part of that is coordinated by Bob Blaisdell, so a former student of Betty. That what we do there is is dry and encapsulate phages for magistral use. You can find this entire Belgian magistral phage system published in open access in viruses, and the phage monograph is supplementary material to that publication. So this is a bit a provocative graphical abstract. So what we did is we took the phages out of this drug development and marketing funnel that takes like on average 10 years and millions of dollars. This publication, the co-authors, you can see people from the Belgian Institute for Health and also the Federal Agency for Medicines and Health Products, which is the Belgian FDA, showing that it was really a joint effort between the authorities and the military hospital. But this is only possible because the Minister of Public Health said, okay, please do this. Because people forget, but authorities like the FDA, they are not there to change rules. They are just there to see that the rules are applied. And without a mandate, they will not change frameworks. So this framework was implemented in 2018. And next step is to get this phage monograph in the European Pharmacopoeia. That way, the system can be used in different countries in Europe. So I will not go into details, but it's all like administration. But now in principle, the European Commission agrees to put the monograph in the European Pharmacopoeia, and there's some committee that will analyze this and discuss how this will be done. So next step is need to go to the patient. And since the implementation of this framework, the number of therapies that we coordinate are increasing. And we have sent phages to more than 20 hospitals in more than 10 countries. And the person coordinating this in the military hospital is Sarah Jibara. She had a talk on the ECMID conference a few weeks ago, and I think it has been recorded, so you should be able to see it. Now, just very short explain these cases that were analyzed. So she analyzed 66 cases. And what you see is that indications were mainly lower respiratory tract infection and bone and prosthetic joint infection. Now, when you look at the treatment outcome, it's not all successful. You can see the yellow is unsuccessful. But you can see also some microbial eradication. Is this kind of gold color, and clinical improvement, light blue. What we saw is that the rate of microbial eradication was highest in bloodstream infection, and then in bone and prosthetic joint infection. But you need to be careful because the numbers are very low. We are talking overall about 66 cases. So for instance, this 80% is 4 out of 5. So what is our experience with this magistral phage framework? So as you saw, we had applications by different MDs in different hospitals, more than 20 in 10 countries. This meant also that we had a large diversity of treatment and sampling protocols. Also, of course, every MD in every hospital wants to publish his own case and wants to report his own case. So you get fragmented reporting. Sometimes I think you will even have no reporting. So we have no patient cohorts, no standardized protocols, or no standardized data. It is impossible to demonstrate efficacy of phage therapy that way. And then we were also confronted with what is called the Arrowsmith dilemma. I think this expression was coined by Scott Stibbits of the FDA, I think 2 years ago here in Evergreen. It's the first time I heard this, term. What is the Arrowsmith dilemma? So first, Arrowsmith is a book written by Sinclair Lewis in 1926. It was considered to be a very good book because it got a Pulitzer Prize, and it was turned into a film in 1931. And also the movie was apparently good because it came second in the Oscars. It's about Martin Arrowsmith, who's a young physician who discovers a phage that destroys Yersinia pestis, which is, of course, the bacterium that causes bubonic plague. Now, what makes this book and movie interesting is that it's based upon the experience of the famous bacteriologist Paul de Kruif, who from the beginning was very much interested in phage therapy. He was well aware, and so it's very realistic, this book. Now, what is the Arrowsmith dilemma? Well, it is, and I was also confronted with this a few times when I presented at conferences, is that there would be someone in the audience that would say, look, you give phages to medical doctors that ask without knowing their efficacy as determined in a randomized controlled trial. So you take away the incentive of those doctors to perform these randomized controlled trials because you already give them the phage. So you should pursue what they say scientific precision through RCTs and not give phages to medical doctors to treat desperate patients. Now, this is dilemma, and I hope the movie will work and the sound also. I will illustrate this with 2 fragments from the movie Arrowsmith.
What is the experiment?
It is agreed that it will be of value to know what this stuff of yours is good for.
Why, yes, of course.
Well, if you could give injections to, say, only half your patients and sternly deprive the other half of them, then you would know.
I certainly would, yes.
That is the experiment, Martin.
Well, I've done that sort of thing with cattle, but good Lord, Chief, these are human beings.
So you're just a country doctor after all.
country doctor be blowed. I've got the bowels of compassion in me. These people are going to be dying and begging me for help.
Man, it's up to you to stand up and take it on the chin.
So at first, he does not want to do this controlled trial, but they persuade him, and so he decides, okay, I will do an RCT, and he travels to the Caribbean and to an unspecified island where they have seasonal epidemics of the plague, and he gives phages as prophylaxis against the plague to half of the people of this island. So randomly, half of them will get phages, and the other half will get a physiological solution. Now, of course, what happens is that the plague comes, and the people with phages survive, and the people with physiological warfare start to die. The problem is that his wife traveled with him, and she didn't get phages, and then she dies.
I loved you, Lee. You knew that, didn't you? Didn't you know I couldn't love anyone else? Didn't you know that, Lee? Go on, give them all the serum. Shoot them full of it. Come on, open the doors. Let them all in. Open the doors!
So I think this is just to illustrate that it's easy to say, you know, you need to deprive desperate patients from a possible treatment when you are at your office, on your chair. And it's totally different when you are in the field and you are confronted with desperate patients and their family. So that's why we say that we should try to have both, to do the RCTs and meanwhile also to use the phages to help treat desperate patients. What are our future plans? Well, we are establishing a phage therapy committee. We already had a few meetings for the coordination and standardization of phage therapy products and protocols. This is very important. We, of course, need to perform the phage therapy RCTs. For that, we need money, so we requested funding. We should implement a centralized database for phage therapy cases that would allow us to, in the long term, to evaluate the efficacy and the safety of phages. There's also work in progress. So already Sarah created a database with 66 cases. This is in collaboration with, again, the Belgian Institute for Health. That's a safety project. But there's also an international initiative coordinated by the Bargrist University Hospital in Zurich, by more specifically by Dr. Shauna McCullen. And we decided not to have two separate databases, so we will integrate into this international database. There's some kind of consortium, some early consortium with collaborators across Europe, the US, and Australia. It's all in progress. First steps are, of course, organizing legal documents. So anyone who is interested to contribute to this database can contact Shauna. Then, of course, we would like to produce phages, but my bosses tell me, no, you are a research lab. It's not your core business to produce phages. So you need to collaborate with industry. So we collaborate with the Belgian company Visal Biosciences, and so they are the ones also that hired Bob Blaisdell, so Betty's student. And so we are starting this collaboration. And then we also want to develop a cell-free phage production system. I will explain why. So we like personalized phage therapy, but the logistics are a nightmare. So what you see here is the transfer of phages and/or bacteria to and from the Millet Hospital in Brussels, and transfer of patients to Brussels. So I spent large parts of my week organizing these transfers of phages, bacteria, patients. So that's why we have developed what I call phage therapy dream. And we would like to realize this for 2035. It's rather complex, but we published this recently. So in fact, what we would like to develop is a device that would produce synthetic phages based on a predicted genome, and a genome predicted by artificial intelligence. And this genome would then be synthesized and then expressed using cell-free TxTL systems. It would be based also on some community effort to obtain phages, and it would be kind of organized by distributed ledger technology. And you would imagine like some kind of phage coin that you would use to exchange services or phages. Next month, we will start the SynPhage project, which will try to get a proof of principle for part of this dream, and it's the part that will predict genome, will synthesize it, and then will express it in a cell-free system. I would like to thank my colleagues. I also would like to thank you. Also want to invite you to the Phage Club later today. It's a clubhouse. It's hosted by Sabrina and Adriana, who are both here. And if you want more information, you can contact me. Thank you.
So questions from this audience?
That was really wonderful, Jean-Paul. Thank you. What I'm wondering is what options or what pathway do you have when you receive bacterial strains from a patient that are not susceptible to the phage that you already have purified and approved for use?
Yeah. Well, it depends also on the time. Huh? So if we have time and we see some moderate activity, then the two researchers, Maya and Thea, will try to train. But this is not even a moderate activity in the phages that we have in the bank. Then it's possible to go to the environment and to get new phages. And we have been allowed in the past to, if we find them, to use them in patients without having the genomic passport and all, because it takes a lot of time. Huh? So you have to do a risk analysis. Huh? But of course, this is like one time, huh? Because after that, you need to promise that you will do the genomic passport and to do everything that is necessary. But it's a risk analysis. You know, the patient is dying, you find a phage that is active, and they are kind of flexible. Like even the training is possible without calling this a new phage. You know? The trained phage can be applied based on the genomic passport of its ancestor. But that's why you also have this banking system. So you cannot have like a drift forever of these phages. You have to be able to go back to the banked phage.
Are there any other questions from this audience before I go to the questions? Okay, so I'm going to pick a few because there are many questions. The first one is from Bruce Levin. It seems clear that if phage therapy is going to be broadly used, it will have to be in combination with antibiotics. It would be unethical to do phage therapy only when there are effective antibiotics. You can comment on that.
Yeah, I agree. Yeah, because the thing, antibiotic resistance. What is antibiotic resistance? You get an antibiogram, and sometimes you see, okay, resistant to all antibiotics. But this is arbitrary because susceptible, intermediate, or resistance is based on an arbitrary cutoff. It doesn't mean that there's no activity. It's based on minimum inhibitory concentrations, and someone will say, no, this is the threshold to call it resistant. So there's still some activity, and then you can clearly see that when you have good combinations of antibiotics and phages, you can get synergy. So yeah, I think I agree. I think to use them together with antibiotics would be the way to go.
The next question, how are you trying to avoid a problem of contamination of lytic phages in therapeutic preparations with temperate phages released from the lysogenic strains?
Yeah, this is what this institute that does the approval of the phage preparations is looking for. So they will look for also for contamination with other phages.
And this is a question from Michael Shamash. Are the genomes of approved phages in the bank regularly reanalyzed as we continue to learn more about the different types of toxins and lysogeny proteins as the public databases improve?
No. No, but I think this is, now this, I talked about the safety project, and I'm quite sure this will be part of that, because now at this, so this Belgian approved laboratory is a former public health institute. So it's not a company, it's a public institute. And they have now a dedicated project for everything that is phages. It's called safety. It will also look at maybe which other toxins we should screen for, not the genes, but the toxins themselves in those preparations. So it's an entire project, and I'm sure. But you need to know this system exists since 2018. So, and we didn't immediately start to treat patients in that system. So we're talking about a short period.
Scott Stibbets would like to point out that the modern ethical standards do not allow denial of standard care as proposed by the stern gentleman in the Arrowhead movie clip. That was a very
Yeah, I know Scott, he also mentioned this dilemma in an Arrowhead movie, but we didn't have the exact same vision on that.
I think we're good. I think we covered most of the questions. Thank you so much for accepting this invitation and coming over and flying all the way over. Okay, so we'll continue with the rest of the now third session of the day. We welcome Christian Harrison. Christian, are you there?
Yes, I am here, sorry.
Okay, perfect.
Working on two screens.
So next is Christian Harrison from University of Leicester.
I'm hoping you can see my screen now.
Yes, we can. Just
can you see the proper view or the presenter view?
No, we're seeing the presenter view.
Okay. Can you see the proper view now?
Yes.
Awesome. Okay, ready. So hi, I'm Christian Harrison. So I am a third-year PhD student with Andy Millard at the University of Leicester, and I'm going to be telling you about the work I've been doing during my PhD project on engineering bacteriophages to combat antimicrobial resistant bacteria, or as it is affectionately nicknamed, murderphage 2.0. So the overall objectives with my PhD were to genetically engineer a phage to carry a payload that would help increase its virulence against bacteria and overcome some of the potential pitfalls of bacteriophages as therapeutic agents. So specifically, I've been trying to engineer a bacteriophage to produce colicins, which I'll come on to what they are in a bit. And the hope of this is that I'll be able to increase the virulence of the phage, maybe delay the development of resistance to a single phage, and try and broaden the killing activity of the phage as well. And then doing all this in vitro, and then at the minute I'm trying to move into an in vivo model using Deliria, so waxworm moth larvae. So the phage that I'm using is called SLR96. It's a T4-like phage with incredibly similar genome to T4, so we have been, to all intents and purposes, treating it like T4. So it can tolerate up insertions of a few kb quite stably, which is one of the reasons why it was chosen, because we want to put something into the genome, so it needs to be able to tolerate that. And there is an existing mechanism to be able to engineer this phage already by causing mutations in a specific gene, the AC gene, which confers resistance to acridines. I'm not going to go into too much detail on this because in about 3 talks time, I think Anna from Dominic's group in Canada is going to go into more detail on this. But briefly, when the AC gene is present and not mutated in the wild type genome, and you plaque assay with acroflavin, you get a reduced number of phage out. When you mutate that gene by inserting a gene of interest, for example, then you can select for recombinants in this way. So that's my phage. So what am I trying to put into my phage? So I'm trying to put in colicins. So these are protein toxins produced by E. coli that are active against closely related strains of E. coli. So they broadly, these are just a few examples, and they broadly look like this. So they have a receptor binding domain, which does what it says on the tin, translocation domain, which helps the colicin move through the membranes into the actual cell, and then the cytotoxic domain, which actually exhibits the killing function, which is most commonly either as a nuclease or with has pore forming action. And they have been proposed as an alternative to antibiotics themselves, but have similar limitations to phages in terms of not particularly broad host range, and they're quite difficult to produce in a lab at scale. So, brief overview of how I want my engineered phage to work, or what I think it should look like at the end. So we all know how a phage infects. It binds to a receptor and injects its genome into the bacteria. It then starts to replicate, making more DNA, more phage particles. I'm hoping that my engineered phage that will produce the colicin as well during the phage infection, so that when we get to the end of phage infection and the phage lyses the host cell, it releases new phage particles and lots of colicin as well. So say that this phage comes up against a bacteria that it can't infect, it doesn't have the right receptor to bind to, so it can't infect that. That phage will wander off and go and happily look for another susceptible bacteria. However, this phage-resistant cell may be still susceptible to the colicin because the colicin is targeting a different receptor. So the colicin should be able to bind to the receptor, translocate in, and then kill this cell that was resistant to the phage. So this, So this should mean that we're increasing the pressure on the bacteria to become resistant to 2 things as opposed to just one thing, phage. So it needs to become resistant to both the phage and the colicin, provided that there are still susceptible hosts there for the colicin to be produced in as well. So the work I've done so far, before I get on to some of what I think are interesting results. In brief, is cloning the colicins into a homologous combination vector. I would like to stress this was not a trivial thing. Cloning toxic genes against E. coli in E. coli is quite tricky and frustrating. They do come with inbuilt immunity proteins to help mitigate this, but still a bit tricky. So I was working with 2 different colicins. One was a nuclease, one was a pore former. The one that I'm going to be talking about is the pore forming colicin, as this one has been working best. The nuclease one didn't really work very well. So then after they were cloned, they were recombined into the phage genome using the selection method previously mentioned. And then I confirmed that the engineered phages were producing the colicin and producing functional colicin. And here are some of the results. So what I wanted to know was, is there an increase in virulence? So this looks a bit messy. It will make more sense. So SLER96 is my wild type phage. Murder phage 2.0 is my engineered phage. So I was using the virulence index developed by Dom's group, Storms et al. 2020 paper. If you've not read it, great paper, go read it. So basically I infected my bacterial cultures with different MOIs of both phages, and then saw overall the MOIs, which phage was better at killing. So this distills down into this, where I've looked at each individual MOI and they've assigned them a local virulence index. So here is the theoretical maximum virulence index of 1 over all MOIs. And as the MOI increases, you can see that they both get better at killing. They both get more virulent as the MOI increases. But the murder phage 2.0 in purple seems to be consistently higher, have consistently higher virulence indexes at all MOIs than SLER96. This indicates that there is an increase in virulence there, and this can be distilled down into a single number. So this is the virulence index across all the MOIs. So for SLER96, it was approximately 0.4. For murder phage, it was approximately 0.51. So it's an increase of 0.11 in the virulence index, which was shown to be statistically significant. So my engineered phage does have an increased virulence. It is better at killing the bacteria. This is also demonstrated by another measure called the MV50, which is the MOI at which 50% of maximum killing capacity is achieved, similar to an LD50 for drugs. So if I go back to one slide, so it's this dotted line here that we're looking at, and the MOIs of each of these phages at which this 50% capacity is achieved. So the MV50 of the wild type phage was approximately 10 to the minus 3, whereas the engineered phage, it was 10 to the minus 4. So the whole log drop in the number of phage that you need to put in to achieve the same amount of killing of the bacteria. So in theory, this should work in, okay, so what am I trying to test? Does it work in co-culture? So if I have 2 bacteria cultured at the same time, one is susceptible to the phage, one is resistant to the phage, mix them together and infect with my wild type phage, does the phage resistant cell die? So I was using one of the mutants generated by Lucy Gannon, who you heard from previously, to see, and mixed it together with my wild type host that was actually resistant to the colicin. So when I mix these 2 together and grow them in the presence of no phage, they coexist perfectly fine. Neither is out competing each other, they just bumble along fine together. When you infect with the wild type phage, the phage sensitive cells in blue take a massive drop off a cliff and get killed by the phage, but the phage resistant cells, as expected, are left alone. But when I infect with my engineered phage, you get the same drop off from the phage sensitive cells, but the phage resistant cells take a more gradual decline. So why this is quite so gradual, I'm not too sure, but it does show that the phage resistant cells are being killed as long as the engineered phage has a host to propagate on to produce the colicin. And the last thing that I kind of want to look at, I've been trying to look at, and this is a bit unfinished, is how quickly does resistance develop? So as I mentioned earlier, the selective pressure should be higher because there's 2 things to mutate and become resistant, because there's 2 things to become resistant against. So I've not put numbers on this yet, but I'm kind of looking at the distance between where the phages start to plateau the bacterial culture and then when they start to rise again. So the length of time between these 2 points should give us an indication of how quickly does resistance develop. And for some MOIs, it does appear that the engineered phage takes a little bit longer to develop resistance, which is a good sign. So to sum up from my objectives, I have managed to engineer a phage to produce, and it does produce colicins. It does appear to be better at killing in vitro. Still working on exactly how quickly does resistance develop. And then I will be moving into Galeria models at the minute, actually, and that's what I'm doing at the minute. So is there an increased efficacy of the engineered phage in an insect infection model using Galeria? So that's work going on right now. And I've also engineered a second phage, which uses a strong phage promoter to try and boost the expression of the colicin. Whether or not this will increase its efficacy, I don't know. That's yet to be examined. I'll be repeating these experiments with the new phage as well. So finally, acknowledgements. Thank you very much to Andy for giving me such a wonderful project to work on for my PhD, and my other supervisors, Martha and Nick. Everybody in our lab that's just really supportive, technicians that deal with my spur of the moment. I need to order more restriction enzymes. Dom's group for their advice on the AC method, and everyone else that's given me help during my project. So I'll take any questions now. Thank you.
Thank you, Christian. Are there any questions here in the audience?
Hi, great work. I am wondering what you know about the genetic stability of your insert, because typically when you clone something deleterious to bacteria in phages, they try to get rid of it.
Yeah, so like I say, this is inserts into this phage are generally quite stable because the packaging mechanism, T4-like phages actually package about 103, 104% of the genome. So there is a bit of wiggle room in how much you can put in there before it's detrimental to the phage. My inserts have been stably maintained. I've done several propagations of them and they're still there. And people before me have also managed to stably maintain inserts of a few kb in there as well. Again, Anna will be talking about this in a couple of talks' time, and she might have more information on that for you as well.
And there are a couple of questions here. I'm going to put up one or two of these. Christian, does the production of colicin by your murder phage negatively affect the titer of murder phage when you're making the preparations?
So anecdotally, a little bit. I'm going to be in the next couple of weeks doing some one-step growth curves to assess whether the burst size and lysis time is affected as well. Just from having to propagate the phage, I tend to get a small decrease in the actual titer at the end of it, but nothing terrible to worry about. Like I'm still getting 10 to the 9, 10 to the 10 every time I do a propagation.
And there's two people that asked a similar question, so I'm going to ask a summarize of this. Is there any known virulence or fitness cost? Is your infection kinetics affected? With the presence of
I have data, but I've not analyzed it properly yet. I think there is a slight cost to the pure virulence of the phage because of the production of colicin. But overall, when it produces colicin, I'm seeing an increase in virulence.
Okay. I encourage you to look at the rest of the questions. There are a bunch of questions in the chat. There's also a Q&A session on Thursday in the morning. If any of these questions deserve a full, big answer.
I'll be there.
Probably they do, which probably they do. Next, we have James Van Leuven from University of Idaho.
from the University of Idaho. Thanks for having me. I'm excited to be here. I'm looking forward to chatting with you about the effects of making synonymous changes to the genome of the bacteriophage Phage 174. So the 20 amino acids are encoded for, in the standard genetic code, the 20 amino acids are encoded for by 61 codons. This leaves room for organisms to use synonymous codons at different ratios, and they do. So for example, in human protein coding sequences, CUG is used 40% of the time to encode for leucine, while CUA is only used 7% of the time. And this is not even the most extreme example out there. The factors causing these skewed ratios of codon usage are fairly complicated, but can generally be boiled down to two processes: mutation and selection. Mutation can cause uneven ratios simply because the 4 nucleotides are not equally mutable, and also polymerases have individual biases that can skew codon ratios away from equal ratios. Selection can act on many synonymous features in protein coding sequences. Some of the probably features that are most strongly selected for include translational dynamics. So in many organisms, there's a strong correlation between the frequencies that a codon is used and the absolute abundance of the cognate tRNAs in the cell. And this is thought to be pretty beneficial for translational efficiency. So if there's lots of tRNAs, you can translate the codon fast. There's many other features that can be selected for. For example, overall GC content, bacteriophages, the presence of restriction sites, the folding stability of mRNAs, etc. And the selection on these features can actually be pretty strong sometimes. So since viruses use their host cell machinery, you might think that the biases in virus codon usage patterns would match their hosts. But lots of people looked at the genomes of viruses and hosts, and sometimes there is concordance between these two, especially for viral proteins that are highly expressed, like capsid genes. But just as often, there's no concordance between virus and host codon usage patterns. And so I think this leaves an opportunity to learn a lot about how viruses use synonymous codons. So another way to study this is to go and change virus genomes synonymously and see what happens when you do this. And this has been done in quite a few viral systems. I'm listing most of them here. And the effects can be really large. So if you take a gene in a virus and recode it only synonymously, you can completely kill the virus if you want. Usually you don't want to, but you can have, the point is you can have large effects. So this is a summary of all the many of the studies that have done this, and there's a couple of, I think, things that we want to look at and improve this type of work. So along the bottom x-axis here, I've listed a few of the features that the selection might be acting on. So examples, GC content, codon preference, codon pair preference, etc. And then the dots represent whether or not these papers actually look at these features. And you can see that rarely are there many of these features that are looked at in these studies. And these features can be confounding. So GC content can be wrapped up, for example, in codon biases. And if you only look at one, it may be hard to disentangle them. Another thing that we think we might be able to prove on is, you can see there's little replication done. So all these blank rows mean that there was one construct made in that virus. So pick one gene in the virus genome, recode it synonymously, usually it's by de-optimizing it, and then measure the effects. And there's a few cases here with the numbers where replication was performed, but not a whole lot. And so we designed some studies to try to improve these two things. and learn about codon usage in viruses. We're using the bacteriophage Phage 174, so we developed a pretty simple cloning system where we just took pieces of the Phage 174 genome, clone them into plasmid vectors. We can do a bunch of editing in the plasmid vectors or have genes synthesized and clone them in, that kind of thing. And then cut them out of the plasmids, ligate them back together to make viable viruses. And this is pretty quick and easy. Some of the genes in Phage are toxic when cloned into E. coli cells. So to overcome that, we just cut up the genes into pieces. Here, for example, I'm showing gene A cut up into 4 pieces. So these fragments are individually cloned into plasmids. And then we can recode them and join the recoded fragments either by themselves with wild type fragments or with other recoded fragments. And here I'm just showing an example of the de-optimization of the codon. So at every site, you take the wild type Phage codon and you recode it to the least commonly used codon in its host genome. And that's called de-optimization. Kind of handy feature about this ligation method is that you can just put whatever fragments you want together. So here I'm showing fragment 1 and 2 ligated together, 2 and 3 ligated together, and so forth. And so we generated just all the within gene combinations of recoded fragments for this study. And we extended that to all non-overlapping coding genes in the Phage 174 genome. So here I'm not showing the recoded fragments, but just imagine what's shown in A for all the genes. And we just made all those viruses and then measured their fitness. And what you see is on the left here is the fitness. This is just growth rate of the recoded viruses with 1 recoded fragment in them. And you can see the effects vary quite drastically. So for gene H, there's no effects for any of the single pieces. For gene G, there's large effects, even with just 1 fragment. On the right, I'm showing what happens when you start combining fragments. So the shaded blocks show which fragments have been recoded. And as you move from left to right, it's just an increasing number of fragments. So by the time you get all the way to the right, the full gene is recoded. And for most genes, for gene F and H, you can see that it decreases pretty much linearly, although statistical power is a little bit light here for us to actually say if it's linear. For gene A, there's some interesting things going on. We observe synapse stasis. So this is when you combine recoded fragments, the fitness actually increases from constructs that have less recoded gene, and we don't really know why this is. One thing about this study is, like a lot of other people, we didn't really take into consideration other features that selection might be acting on, so we just de-optimized these without really looking at anything else or paying attention to anything else. So we codon de-optimized them. So a priori went back and looked for other synonymous features that correlate well with changes in fitness. And we found that one of the best correlating features is the stability of the mRNA. So if you recode the gene, and keep in mind this is codon de-optimized, changes in the folding stability are pretty predictive of changes in fitness. And so we wanted to explicitly test this. So we designed another set of experiments with additional recoding methods and some other stuff I'll talk about in just a sec. So again, we did the de-optimization scheme here. We also included random recoding. So this is where you just take a codon and you randomly pick some other codon and change it to that. We also use the shuffle method, in which you just exchange synonymous codons within the gene. This has a nice feature of not changing the overall codon usage of the gene at all. You're just shuffling them around within the gene. It also has kind of a cool thing where you can make a bunch of these shuffled constructs and then run them all through a folding mRNA folding prediction algorithm. And then we pick ones that have large folding effects. So you have the shuffled construct that should have no folding effect, and you pick some that have high folding effect or positive folding effect and negative folding effects. And we generated those. To add replication, we varied the amount of the gene recoded. So we did 25%, you just picked 25% of the codons at random, 50% of the codons at random, 75% of the codons at random, etc. To recode. And in addition, we did 3 replicate constructs of every single method. So 3 gene F, 25% random recoded, 3 gene F, 25% de-optimized, etc. For everything. And so this figure is just showing where the changed codons are in gene F, for example. And you can see that there's more and more changes as you go down from top to bottom. We did this for gene F, G, and H. We also measured fitness using 3 methods. So the standard growth, flask growth assay, as well as a competition assay where we take all the variants, put them in a flask together, grow them up, and then measure the frequencies of the individual genotypes over time using just packed biosequence across the genome, because it's a little genome. And then look at frequency changes. And then as well, we measured plaque size for all the variants or for all the constructs. And these things are pretty well correlated. So we'll just show one, which is the flask-based growth assay. And this is the fitness for all of the constructs shown in this one graph. So it's fairly awful to look at. But you can see some general trends here. Like before, as you add, as you change more and more codons, the fitness tends to decline. There are large differences between genes. So the effects of gene H are fairly subtle compared to the effects on gene G and F. So gene H are these blue triangles here. There's kind of a weird result, which is if you look at the bottom, gene H is actually where we had the hardest time making some of the constructs. So we had a lot of constructs for recoded gene H that we just could not get. And we tried fairly hard to make these. I don't know why that is. Right here, these 3 shuffle constructs are the stable versus unstable ones. And we see no difference, which was a little surprising to us because we really expected there to be some differences when you change the folding stability of the mRNA, but there's no difference between these things. And I think one of the most interesting things is the level of stochasticity between what we thought were replicate constructs. So if you look at in one category, say 75% random, you look at the 3, these are supposed to be constructs that are all 75% randomly recoded, and they're really quite different from each other. And this is pretty much the same kind of story across the different methods. So what we think maybe is happening is there are certain sites in the genes or something that when you happen to hit them, there's big effects, but we don't quite know yet. So one of the things we're working on doing is characterizing some of these large effect constructs using like RNA-seq and proteomics. Another thing that will be interesting now that we can randomly ligate these recoded genes together and create pretty large pools of different constructs that we can measure fitness on using PacBio. And so we plan on looking at how recoded genes combine because we haven't looked at that yet. So that, I thank everybody in the lab and funding support. And it's really fun to be here and talking to people about Phage. So thanks for the invitation. Questions?
Questions from the audience? Questions in the chat? Let me see. I think I'm not looking at all the chat. Are there any questions from the online audience? It seems that there are no questions, but I think it sounds very interesting. changing codons. So, but in terms of evolutionary pressures and what changing codons would do to phages if you put them in the wild or use them later for therapy, how do you expect that to work?
Well, so initially the project was sort of designed to help understand. So there's actually all the viruses, most of the viruses listed before were designed to make attenuated vaccines. And so this in part was to better understand how you can make attenuated vaccines, but there could also be selection on synonymous codons that overcome selection for protein coding sequences. So if selection is strong enough on some of these features, it'll control the sequence of the protein. I think.
Yeah, I studied lambda display. And I thought that lambda was smart enough to optimize its codons, for example, for the D protein. And so I codon optimized 55 different codons. And I found that lambda somehow learned that it's not a good idea to make lots of D. And so this is going on in selection in evolution, I believe. And you're the first talk I've ever heard that really suggests that my stupid observation it may have some merit to it. Thank you.
Yeah, I think, we think there's probably some kind of balance of, some genes are more optimized, some are less on purpose, but I don't think there's a good understanding of it yet. So.
Thank you, James.
Thanks.
There are some other questions in the chat for you. Meanwhile, we welcome John Callis from the Stanford University Department of Bioengineering. John?
Hello, everyone. Give me a thumbs up if you can hear me out in the internet space. I know that the people who are physically in Washington can't thumbs up into the camera.
But I
All
can.
right, sweet,
I'm
awesome.
Right here.
Excellent. Thank you so much for hosting. Yes, thank you. Hello, my name is John Callis. I'm a graduate student at the Stanford Bioengineering Department with Drew Endy. I'm very grateful to be following James because not only have I been following his work and I'm very excited by it, but I also happen to be dealing a lot with Phage 174 and dealing a lot with recoding it and making a lot of very difficult codon decisions in its genome. For context, I'm a synthetic biologist and an engineer. I'm coming to Phage as a mendicant studier. I have no idea how Phage normally works and I'm learning as I go, but I am an engineer and I'm very excited about evolution and evolutionary control. So today I'm going to talk to you about how we can build bacteriophages with reduced codon systems. But first, an homage to Phage more broadly. For 100 years, Phage research has been revolutionizing molecular biology research. And in the last 20 years, phage biology has been revolutionizing synthetic biology, my home field. For instance, the first genome ever synthesized and shown to be viable, synthesized from abiotic chemical parts, was the Phage 174 virus. Refactoring genomes to make them easier to understand by separating out overlapping genes and to put in engineering components that make it easier to manipulate such genomes. This was first done in T7 and in Phage in our lab by respectively Leon and Shri in one case and Paul Jaski and colleagues in the other. And the first genome that we can say that with definitive certainty that we understand its annotation fully, i.e. we know all the genes that are actually being translated and how they're actually supposedly functioning in the genetic system. This first genome to be fully annotated, also Phage 174, and it was done by total genome synthesis where we've removed cryptic open reading frames by ablating start codons and showing really that you can get replicative phage with only the annotated genes. So phage has been a huge motivation and motivating test case for rebuilding phages, but it's also been motivating for rebuilding E. coli and other bacteria that interact with such phages. For instance, work that was originally published in 2019 and then further developed in this year, 2021, has developed a 61 codon E. coli. This is an E. coli whose entire genome has not 64 codons like in the standard genetic code, but 61. Here we can see 2 serine codons and 1 stop codon removed from the genome, and the associated translation machinery also removed. So these ribosomes in this E. coli don't recognize CA, UCG, or UAG codons. And as a result, phage genomes that naturally contain these codons simply cannot replicate. Transcripts with those naturally occurring codons have the ribosome stall on them as the ribosomes seek to incorporate amino acids that it simply cannot incorporate. So as you can see, this gives natural phage resistance, at least in the liquid phage lysis cultures. So phage has been a huge motivation for us synthetic biologists, great test systems to work in, and I'm very excited to be talking with some phage biologists today. What I'm personally interested in are these reduced genetic code systems, these genetic codes that don't have 64 codons accessible to them, but rather fewer. So in 2019, we first reported on a family of reduced codes that only use 1 codon per amino acid. We call these reduced 20 codes or red 20 codes. There's around 700 million different variants of such a code. You just shuffle around the genetic code in a bunch of different ways. But all of these genetic codes have some very interesting properties, much like the work that I just showed you previously out of Jason Chin's lab with 61 codon E. coli. Such genetic codes would be phage resistant naturally. They would prevent horizontal gene transfer as well. Genes that are naturally encoded just simply wouldn't be translated by such genetic systems. But in addition to that, we showed that such organisms so encoded would be naturally resistant to evolution. They would evolve more slowly. So the idea here is if you engineer the genome of such an organism to only use these 20 sense codons, and a mutation occurs, that mutation is far more likely to become one of these null codons for which there's no translation machinery. Ribosomes would then stall on such transcripts, these mutated transcripts, and not be available for translating other proteins in the proteome. As a result, there would be a slight weak fitness effect against that organism and its entire lineage, and that lineage will be slowly selected against over time. So this effect is independent of the gene that's mutated. It's independent of the host organism that's engineered. This should be it should just work across the board for all biological systems. So we found this is a very exciting proposition. And moreover, we actually were able to build a prototype of such a reduced genetic code in a tube. So a cell-free expression system built up from individual recombinantly derived proteins called PURE was used, lacking tRNAs, and we then synthesized 21 tRNAs chemically that corresponded to the red 20 genetic code shown on the right, top right. We combined PURE and these 21 synthetically derived tRNAs to produce a genetic system capable of expressing red 20 encoded genes and incapable of expressing naturally encoded genes, as you can see below. Bottom left, you can see PURE with its natural tRNA set. It's able to express a GFP reporter naturally encoded. SC says means standard code in orange. And in green, you can see that the red 20 encoded variant is translated in natural systems. On the right, you can see our red 20 prototype, where we have only the synthetically derived tRNAs making the red 20 translation system. Orange, the natural reporter, is expressed at a much lower level, about 50x lower than the red 20 encoded variant. So we can show that we have the capacity to, 1, encode genes in such reduced systems, and 2, create translation systems that are selective for such a encoding, which is very exciting. It's the beginning of a journey towards making an entirely red 20 organism such as E. coli. But we've really only recoded single genes at a time. And from then on, we sought to engineer larger and larger genetic systems. And we decided that Phage is the perfect test system for such a project. The genomes of Phage are very small in comparison to E. coli with 4 megabase genome. Phi-X174, for instance, has like 5,000 base pairs, 5,386, and 11 genes. But unlike our previous test cases, the genes of Phi-X174 are entirely integrated. All the parts have to work together. And moreover, 7 out of 11 of these genes are entirely overlapped with each other. This represents the hardest problem in red 20 recoding, because when you have overlapped genes that are in different frames relative to each other, you change a codon in one frame, you change the codon in the other. So you have to somehow choose recoding edits that are valid in both frames. It's a very hard problem, but to misquote JFK, we chose to recode the phage, not because it's easy, but because it's hard. And also because we hope that any headway we make here can give people a little bit more confidence in equally hard systems. To date, we've simultaneously recoded 3 red 20 genes in Phi-X174, gene J, gene F, and gene H. And we've shown that we're able to recover plaques via plaque assay, which is very exciting. This work was done in collaboration with Bradley Wright, Russell Vincent, and Paul Jaskey, now at Macquarie University. During the pandemic, when we were unable to come into lab ourselves, and they were able to actually do some wet work. So we sent them DNA across the ocean. It was exciting. Big shout out to them. Thank you very much. We also were able to show that we weren't able to naively recode gene G for reasons given in the previous talk. It's kind of hard to recode gene G. You can't just make arbitrary codon choices. And because of the constraints of the red 20 code, we don't have as much flexibility in choosing which codons we can change to, so we can't get around things like secondary structure, as mentioned, or mRNA stability. What we can, however, do is change the regulatory elements upstream of such genes. So here I'm showing you a protein gel in which we expressed gene G in its wild type encoding and gene G in its red 20 encoding, and showed that the red 20 encoding is simply not making protein in an in vitro translation system, not even in vivo. We hypothesized this might have something to do with mRNA stability or our translation initiation, and not worrying too much about the mechanism as engineers, we tried to get around this. And we did so by engineering the RBS of such a gene to express at a much higher level as predicted by an RBS calculator. And we showed, at least in vitro, that you can recover a slight amount of expression of gene G red 20, which gives us some excitement. We've ordered synthetic DNA to now try whether this RBS edit will recover viable phage. The DNA is now, it was delivered this morning, actually. It's now sitting on my bench directly across from me. I'll let you know how it goes. Follow me on Twitter. However, as I said earlier, the hardest part to recode are the overlapped genes. And it's not just simply difficult to think about. We've actually been able to show using a method called satisfiability modular theorems, or SMT solvers. We've been able to show computationally that it is physically impossible to red 20 encode Phi-X174's overlapping genes without introducing some non-synonymous changes. So the question is, which ones, how much can you change any individual protein in such a virus and still recover viable phage? What you can see here are 2 genome designs that I've made where we've recoded either one of 2 overlapping genes, either frame 1 or frame 2, frame 1 on the left or frame 2 on the right. And that corresponds to recoding gene A, gene C, and gene D simultaneously, while not worrying about what effects occur to the protein sequence of B, K, and E, or the other set of proteins, recode gene B, gene K, gene E, not worry about the effect to A, C, and D. Fun fact, neither of these designs work, which is unsurprising. That's because we're completely ablating the protein sequence of the other sequence when recoding. So we've shown that the naive approach doesn't work, and we are now looking for better solutions. So instead of trying to recode the entire gene's whole hog, our next approach is to build genome variants at large scale and test individual edits at a time. In a wild type phage, make one codon change in one overlapped gene, and then test those in massive parallel. We're going to be leveraging some very cool technology that's only hit the market relatively recently, which is oligo pools synthesized on a silicon chip. This allows you to order hundreds of thousands of unique sequences and have them delivered in a single tube, which allows you to do massive high throughput screens such as these. And then you just build genome variants, transform them into cells, test with next generation sequencing which of those genome variants actually replicate phage. It's a very exciting technology. I'll report back on that. Once again, follow us on Twitter. Taking a step back from Red 20 and coming back to some of the great work coming out of, once again, Jason Chin's lab in Cambridge. They showed that reduced codon systems can be phage resistant. I thought that was really exciting, but then I questioned it. How phage resistant really is a genetic system that is only lacking 2 or 3 codons? I mean, could you just engineer a phage that targets such an E. coli? We think so, actually. The Phage genome only has 38 instances of TCG codons and TCA codons, 19 apiece, and no TAG codons. There are, in fact, no TAG stops in the entire genome. So there are only 38 instances of codons that need to be recoded in this genome such that all of its codons are completely compliant with the SYN61 genome. So we decided to take on this topic, and to date, we've actually been able to recode 80% of the Phage genome simultaneously and recover viable lytic phage. You'll note, so the highlighted sequences in purple are the recoded genes. The sequences in gray are the wild type non-recoded genes. You'll note that the genes I was unable to recode are once again the overlapping genes, of which there are, about 19 edits to make further. So once again, the DNA has literally come in today for at least 7 of these changes. I am going to make all of these changes individually, one at a time, in a wild type background to determine which of these are non-viable. And then, having determined which of these changes are non-viable, we will use the oligo pool method described previously to screen many variants at a time at each individual non-viable position in order to find viable alternatives. It's very exciting work. We're very happy to be working in such an exciting system as Phage, and I welcome your questions.
Thank you, John. Any questions from the audience here?
I see 2 in the chat. Should I just start responding to them?
There's a question here first, and then you can answer the question in the chat.
Hi, Jacques Matthew from Rice University. I was wondering, since you're having a big problem with the overlapping genes, and I guess you're constrained, you can't actually just refactor it and put them in line because you're probably constrained by how much it can package. Is that correct?
Yes, that is correct.
What I was wondering is, why don't you use a filamentous phage such as M13 there you go.
That's a great question. So, I mean, that's kind of, okay, it's not hush work, but that's definitely been some of our back burner work. Yeah, M13 is awesome that you could just extend its genome indefinitely. Well, not indefinitely, but like 4x its size or whatever. However, I perhaps I didn't articulate this as well when I was bringing up why Phage is such a cool test system. In E. coli and in other bacteria, in fact, in humans, you have overlapping sequences. So in a lot of natural systems that we would like to red 20 encode, there exists overlapping sequences. And while refactoring is a great solution around such a problem, we wanted to show, if possible, that one could just recode overlapping sequences, and that shouldn't be a consideration when using such a technology.
There are like 3 questions in the chat. I would urge, because Manoj, I know Manoj's work. He did work on RZ1, untangling RZ1 in Lambda, and it's precise. It's super cool work. But there's a question from Brian Gibb. It says, can you push evolution by reducing tRNA levels to target codons?
I think, yes, I understand this idea. So what you're proposing is to reduce cellular levels of a tRNA to lower and lower amounts such that the genome naturally depletes those codons. Yeah, that's a really cool approach. I think it would actually get you really close to it, like a very cool starting point. I don't see why that wouldn't work. However, I will say that perhaps as a stylistic opinion, our lab is one of rational design in synthetic biology. And while we believe that the directed evolution approach is super cool, the kind of work that we're taking on here is hopefully one in which we learn design principles such that we don't have to rely on directed evolution for such approaches. I think that's the right way to do it, to be perfectly honest. Yeah, but if we're here to learn engineering principles, then I would propose perhaps a rational design approach, or like a spray and pray approach that gives us more knowledge about the underlying biology.
Thank you, John. There are 2 questions that I encourage you to answer in the chat. We will continue with Anna Avizu from the University of Alberta. Anna, are you there?
Yes, can you hear me?
Perfect.
Okay. Can you see my slide?
Yes.
Awesome. So hello, everybody. Thank you so much for the opportunity to speak today. I'm going to talk about a method developed by our group for phage engineering and how we have explored its efficiency and how to improve it. But first, why should we engineer phages? Well, phage engineering is becoming more relevant because it speeds up the development of phage-based technologies and the expansion of their applications. However, unlike in bacteria, current methodologies are limited, and that is why we need to grow our toolkit and be able to overcome those limitations by saving time and avoiding non-functional phages or inserts. Current problems are behind the motivation or need for phage engineering. The rise of foodborne illnesses worldwide makes 6 million people sick every year, which translate in 1 in 10 people getting sick. And from all the deaths, 40% account for children younger than 5 years old. This problem alone in the US is costing $36 billion every year, and it's increasing. Additionally, by the year 2050, infections caused by antibiotic-resistant bacteria could reach 10 million deaths globally. Having more options on the phage engineering toolkit can contribute significantly to solving these issues. Over the years, several methods have been developed with the aim to modify phages for different applications. A lot of the current methods rely on homologous recombination to engineer phages because it allows us to introduce foreign DNA into the phage genome by flanking the sequence with regions of homology. However, this mechanism by itself is limited by low recombination efficiencies. And without a selection method, the recovery of recombinants can be very difficult and long. Also, it has limited scope due to the unknown function of phage genes. After the hurdle of finding a potential phage for an application, Some of the methods available for the modification are time-consuming, costly, frustrating, and more often than not, lead to a dead end. However, several strategies have been developed to overcome some of these issues. One example is bacteriophage recombinant electroporated DNA, or BRED, which is an in vivo method where the phage DNA and the insert of interest are co-electroporated into electrocompetent host cells. Recombination takes place by relying on homologous recombination. However, to recover successful recombinants, we require highly competent cells, and this approach for lytic phages has low efficiencies. The screening process is usually very long, but can be done in combination with CRISPR as a control selection method to speed up the process. Another method is the cell-free transcription translation systems, which offer a high level of control and freedom over in vivo methods. It allows us to reboot phages without the need of the bacterial host. This method was used to produce T7, T4, and other type of phages successfully. Yet, it isn't known if it's universally applicable, mostly because of the different phage assembly mechanisms. I'm sorry, my computer is acting out today. Although these and other strategies solve some of the hurdles, two issues remain. We require a lot of information to design new phages, and we still need to screen for the modified version. The goal is to engineer any phage that we want without needing full characterization. To achieve it, we can build a cassette that allow us to accelerate and facilitate modifying our phage efficiently. With this rationale, our group is using the ACT gene present in the T4 phage genome as a door to engineering. During the 1940s, researchers working with phages noticed that acridine, such as acriflavine, impede the replication. In the 60s, the ACT gene was identified. And during the 70s, it was thoroughly studied. However, after that decade, not much was done, but a vital piece of information from this research serves as the foundation of our method. And that is that demodification leading to an unfunctional protein or deletion of the gene makes the phage resistant to acriflavine. So we can replace the ACT gene with a cassette where a reporter gene or another gene of interest is flanked with two regions of homology with the T4 phage genome. Then the phage will infect the bacterial host current a plasmid with our construct. I'm sorry, I think something happened because my PowerPoint just stopped.
Yeah, Anna, so what you can do is actually stop your video and just play the PowerPoint. Maybe that will help with your connection.
Okay. Thank you. Thank you for the tip. I'm sorry. I'm sorry, my PowerPoint.
Yep. Are you able to share it again?
Give me 1 second.
Yep.
technical difficulties that happen when you're doing this live.
Yes.
It's amazing we've gotten this far without more of this.
I know.
You see
There
my
you go.
slides?
Looks
Yes.
Okay, so then after we design our expression cassette, the phage will infect our bacterial host, in this case E. coli, and after homologous recombination, we will get new phage progeny. Then the resulting lysine is plated onto selection plates. Because of naturally occurring mutations, some wild-type phages are resistant to acroflavin. But if you look at these pictures, the background is not that high, making the selection of recombinants an easy process. After establishing these, we moved on to the optimization of the method to find its efficiency, versatility, and limitations. First, we evaluated the impact of the length of the homologous sequence. We performed experiments with 3 different lengths, 55, 100, and 200 base pairs, being 200 base pairs the best option. And with this data, we established that this is the primary determinant of recombination frequency. We also tested the recombination efficiency based on the homology length using 2 approaches. Direct selection from recombination with a 45% of efficiency, while selection after amplification had a recovery of recombinant phages higher than 95%. In summary, with 200 base pairs of homology, we can modify and retrieve engineered phages in a couple of days with more than 95% of recovery. Once we established our method, we investigated different variables to improve the design of the inserts. We looked into the GC content and the insert size. The GC content is crucial because the T4 phage genome has a 35% of GC content. So we wanted to know if it affected the recombination efficiency. We managed to get recombinants up to 62%, but by reducing the GC content, we got a reversion frequency over an order of magnitude, and this was confirmed with several genes. And regarding the insert size, 2,500 base pairs is the limit to recover successful recombinants, since 4 kilo base pairs didn't result in a higher reversion frequency than the wild type. The length of the insert likely has an impact on the packaging capacity of T4. Using the nanologue gene, we designed different cassettes with different promoters and RBS and measure luminescence. The construct with the G23 promoter, which is the cassette at the bottom, had the most bioluminescence expression. These cassettes that have different levels of expression, we found by measuring the specific luminescence per phage count that the construct do not affect the amplification of the phage. Another important question is if the inserts affect the ability of the phage to recognize and kill their host. To assess this, we use the virulence index. The test is performing a 96 well plate at different MOIs. After plotting the resultant curves, we compare each of the death curves against the control. Integration of both curves up to the onset of a stationary phase using this equation give us the local virulence at a given MOI. The smaller the area under the curve, it means the more virulent the phage is. Then we plot the local virulence versus the log of the MOIs tested, and we integrate a curve using this equation, and we get the virulence index, which is a quantitative measure of virulence that can be used for comparison. Using this analysis, we compare 4 different engineered phages carrying different cassettes against the T4 wild type. And we found that their ability to kill their bacterial host remains the same even after modification. Different genes and the presence or absence of promoters do not affect the virulence of the phage. We also tested if the method could be used in phages other than T4. So we found that T2 and T2-like phages carry the PR gene, which makes them susceptible to proflavin, another type of acridine. The ACT gene and the PR gene have high levels of identity. So we chose PPID01, a T2-like phage specific to E. coli O157H7, and we modify it using the ACT method by introducing the nanoleukocyte into its genome. As you can see with this graph, we engineered PP01 successfully. By using this engineered PP01, we developed an application. We loaded a plastic material, electrospun PHB, with the engineered PP01 phage by 4 different techniques, and we got a really good loading efficiency. Then we measured the infectivity of the phages present in the 4 materials, finding that the electrospun fiber mat had the best response. Using this material, we developed a swab and tested it on TSV media and contaminated milk. With only 1 million of exposure of the swab, we detected the pathogen at 10 to the 1 CFV per ml just after 1 hour. This work is a stepping stone that opens the door to phage-based technologies, such as a smart swab test for the rapid identification of supershredders in cattle, and to monitor cleaning practices of high-risk areas where food products are being handled. In the healthcare system, we can identify bacterial pathogens rapidly and monitor equipment and surfaces of these environments. With this technology, we can develop a smart food packaging to detect pathogens throughout the distribution chain, minimizing the risk of foodborne illnesses. In summary, with our method, we are contributing to the current toolkit and pool of technology available for phage engineering. Additionally, we've gathered valuable information about the design of inserts and cassette that is applicable not only to our approach, but to other methodologies as well. And lastly, we have developed an application for the detection of a pathogen in food. And with that, I'd like to thank the funding agencies and members of my research group that made this work possible, and the organizers of this event for giving me an opportunity to present our work. Thank you.
Thank you, Anna. Are there any questions in here? There are not questions from the audience? Okay, there's one question from the audience.
Thank you, great technique. I am wondering what you could tell us about the presence of AC-like genes or PR-like genes outside of the TEEV and phages.
Well, with our literature research, we found that T4 like T2 and T6 are the ones carrying this gene. We haven't found other type of phages carrying these type of genes, to be honest.
Okay, I will encourage, there's questions for you in the chat that I will encourage for you to answer, and so that we can continue. We have now Catherine McGinney from Sandia National Labs. Can you share your screen?
I did share my screen.
No, I cannot see your slides and your co-host. Let me see. So we're seeing you, but not your slides.
I think you need to start video on your end, Catherine.
All right. So it may not let me.
Okay, if not, then I can play the slides for you
Can
up
you?
here. Yes. Okay.
So you're gonna put them on your Zoom screen,
Yes.
Adriana?
Yes.
We jinxed ourselves with the tech.
We were so close though, so close.
This is nothing. It's all good. Do you have any jokes to tell, Catherine?
No.
Me neither.
Okay. See if this makes sense. Okay. And now you're supposed to be sharing the screen. We tried it. Did it work? No, for some reason, it's not liking me. I'm sharing the screen.
So you're sharing, but it looks like it's sharing from your end?
So it looks like it's sharing from my end, but it's not. So for some reason, it's not liking me.
Maybe stop and try again.
Yep. Stop share. Let's see if I can share it. Let's share it from the Evergreen here. Okay.
Wow, yay.
Thank you. All right, so thank you to the organizers for allowing me to speak today. Today, I'm going to talk to you about a computationally guided universal phage engineering platform. Next slide. So here's just a brief outline. I'll talk to you about our prophage identification software, how we use the software to guide phage engineering, and then the application of our whole platform to Berkeley area. Next slide. This audience already knows, but phages are found in nearly every environment. They have a lot of broad impacts and a lot of applications. But one of the things missing is large-scale identification of phages, and more synthetic biology techniques are really needed to generate a phage for every bacterial species in every environment and for every application. Next slide. So the current way is we go out and we find phages from the environment, and currently in GenBank, there's about 15,000 unique bacteria phages. The way that we think about this question is that bacterial genomes are really nets that catch phages. So these phages are typically more host adapted because they're chosen from close relatives of the target species. And we look at our prophage prediction from a small scale study of about 2,000 bacterial genomes. We predicted 1.2 prophages per genome, and we multiply that by all the current bacterial genomes in GenBank. We anticipate we'll discover more than 400,000 bacteria phages. Next slide. So this really starts with our software that discovers genomic islands. So that's that larger class of mobile elements that contain an integrase. And it discovers them precisely. And it does this using the integration event where we can detect at L and at R sites precisely. And we do this through two complementary software. Islander, which is shown down on the bottom left, looks at tRNA genes, looking for this genetic signature. And what we learned from this was that the integrase gene is typically located near one end of the genomic island. And using that information, we built Tiger that uses ping pong blast approaches in uninterrupted reference genomes to discover the precise at L and at R sites of any mobile genetic element of which prophages are a subclass. Next slide. All right, So why does precise island mapping actually matter? So if we look at the biology of precise island mapping, it allows us to detect phage host ranges. So if we discover phages in the environment or we discover phages in metagenomic data sets, we can compare back to this database and say, here's where the phage actually resides. In a paper we recently published in 2020, we revealed a number of cases where cells use genomic islands and prophages to control gene integrity, especially for things like sporulation, where when the island isn't integrated, that gene is disrupted, and when it's excised, that gene is active and sporulation can occur. And we're currently working, back one slide, we're currently working on looking at satellite phages that are actually integrated directly into their helper prophages. So we have a satellite that's integrated into a capsid gene, and we have experimental evidence that when that excises, the helper prophage can actually begin to replicate. And there's a number of applications that this precision allows. The first is genome editing. Because we have the integrases and their diverse sites specified, we can use that large database that we have. And then what our real ultimate goal is creating a phage factory to generate a set of phages for any target bacteria. And we've already done this in Pseudomonas aeruginosa in the therapeutic sense, deleting integrase genes and using them against Pseudomonas aeruginosa PA01, and that was also published last year. And now we're turning more to microbiome control and study, so looking at phages to kill various members of certain microbiomes and looking at phages as vectors where they can carry payloads to a specific target of the microbiome. So when we apply our software, nope, go back one slide. Yeah, you're a slide. When we apply our software to all the bacterial and archaeal genomes that we find at GenBank, we've currently done about 135,000, but we are working through the more than 300,000 genomes at GenBank, and we're missing Bacillales and gamma proteobacteria, which skew these numbers a little bit to the lower side. We've identified almost 360,000 genomic islands, 75,000 of which are called prophages. We have an accompanying software that actually goes through and detects that, and I'll talk about that in a moment. I'd just like to point out with the genomes we've tested, a lot of these genomes have no hits for islands with integrases, and a lot of this is because there's highly reduced genomes or insect symbionts where you don't typically find them. Next slide. And we break this down into various phylogenetic groups. We see with the yellow bar there that every group has prophages identified and no group lacks them. And again, when we treat Bacillales and gamma proteobacteria, those numbers will be much higher than all these shown here. Next slide. And most of these prophages actually appear to contain a full gene complement. So we have accompanying software that goes in and looks at the length of each of these prophages, what genes are contained, and what we consider that. So most of them contain structural and functional phage proteins. A large number we consider incomplete gene complements or satellites, and we're working to tease those two apart. And then based on some work by others in the literature, we tag innoviruses or the filamentous phages as very small genomes that contain the PFAM-ZOT and or coat proteins. Next slide. So now we're going to apply this into a larger platform so we really can get to our ultimate goal of making a phage for any target. Next slide. So we've done this once where we have a bacterial agnostic phage engineering platform. I've already described this prophage database that we have and it's growing every day. And from that, we can identify prophage-rich close relatives of a target. So we did this for Pseudomonas, as I mentioned, and we found 2 close relatives that had a lot of prophages. We can then validate the activity of these prophages. We have experimental methods like PCR, and we also have more bioinformatics methods, which I'll discuss in a moment. And then you really have 2 avenues. You can induce your wild type phages and isolate those, or you can go through engineering the phages. So we've done long PCR followed by Gibson assembly, and we actually have software that's available to help with those large Gibson assemblies. And then you can reboot in any fashion you like. And what we've currently found is the rebooting is that bottleneck that we're working to get through. So we're exploring various methods for synthetic genome rebooting. Next slide. So in the final few minutes here, I'm going to show you how we've started to apply that phage engineering platform to Burkholderia. Next slide. So Burkholderia was of interest to us for 2 main reasons. The first is it is problematic therapeutically in cystic fibrosis patients, but also Burkholderia is found in a lot of soil microbiomes, so it addresses our second area of interest. And what you're seeing here on the right is a phylogenetic tree of many of the type strains and the strains we can obtain for Burkholderia sapacea complex species. And highlighted in blue are 16 strains that we obtained and tested for all of the prophages and the prophage activity. We discovered 123 genomic islands, 39 of which are prophages, and 30 of these look like those full-tailed gene complement phages. Next slide. Because of the applications that we're thinking about, we really want diverse prophages. So we started using just fast Annie approaches. So when we do that with our 39 prophages from these 16 diverse Burkholderia species, we only cluster into 5 clusters with 21 singletons, suggesting there's a lot of good phages out there for the various applications and that these would be complementary potentially in various cocktails. Next slide. We then like to do a validation on all of our prophages because as they reside in the genome, the bacteria is trying to actively get rid of them. So we can do this through traditional qualitative experimental methods like qPCR. So for this slide and the next one, I'm just going to show an example of one of these phages, but I have all 39 if there's any questions. So this is phage 44U. And because we have those precise ends mapped, we can immediately design primers that detect the APP or that circularization junction or the APB site or that scar left behind in the bacteria. And when we treat our bacterial Burkholderia with mitomycin C, and then we take various time points after mitomycin C induction, what we see is that this phage is circularized in the filtrate at a number of time points post mitomycin induction. And we will note that we see a positive hit in the 0 hours post mitomycin C, which is just the spontaneous induction that's observed occasionally. And we see the complementary thing with the APB primers at 2, 6, and 24 hours post mitomycin induction. We see a scar where this phage is excised. So by PCR, we can quickly determine this phage is excising and circularizing. So it is likely an active phage. Next slide. But we like to give a little bit more quantitative information, so we can do this bioinformatically. We published a paper a few years ago now on a software called Juxtaposer and AtCount. And what we do is we induce with mitomycin C, and then you deep sequence that bacterial genome, and our software searches for the recombinant reads that correspond to the circular and deletion junction. And as you see here on the right, you can actually tell how many bacterial cells have excise a phage and what the replication looks like. So shown down here in blue, you can see there's a low level of excision over the 6-hour time course that I'm showing. And in the gray line that's a little bit lighter on the screen, you can see that at 1 hour, you have a low level of circularization. At 2 hours, that's growing beyond what's just excised from the bacterial cell. And at 6 hours, you have a lot of replication, suggesting that this phage is not only excising and circularizing, it's actually replicating, suggesting it's an active prophage, apt for many of the applications that we're looking at. Next slide. And finally, a little bit unexpected to us, the Burkholderia prophages that we study tend to have narrow host ranges. So I mentioned we did a lot of work with Pseudomonas, and we did this host range study there with a lot of clinical strains, and our Pseudomonas phages infected pretty much everything we tested. But if you see here, on the bottom is all the prophages from each strain. On the top is the filtrate that was tested after 6 hours of mitomycin induction. And down the left side is the lawn that the host was made of. And you see there's a lot of non-green boxes suggesting that that filtrate did not leave clearings and or countable plaques on that plate with and without dications. What we will notice is on the right are all the Burkholderia cepacious strains, and those seem to have some cross-infectivity of their prophages. So this is another consideration when we're thinking about therapeutics and using prophages in microbiome situations for Burkholderia, but we may need to be even more specific than just the genera or the species. Next slide. And finally, just to wrap up with a few conclusions, bacterial genomes are a rich source of prophages, and we're developing a large prophage database. Because we already know the sequences, genetic engineering can occur quickly through any technique of your choice. And this platform can be universal. We can rapidly implement it to make phages that kill or phages that carry payloads like vectors. And finally, we've applied it to Burkholderia, which harbor a lot of diverse prophages, but they have narrow host range. Next slide. And I'd just like to acknowledge the Sandia Phage team, especially Kelly Williams, who did a lot of the development of the software, David Wagner for all those Burkholderia strains, and our funding sources. Final slide. And I'll take any questions. And if you're interested in collaborating on any of this work or looking for a postdoc, send me an email.
Thank you. Despite the technical challenges, we made it. Sorry. I think we jinxed ourselves. I'm so sorry. Are there any questions from the audience?
I was just curious, you said the bottleneck there was in your rebooting.
Yeah.
Is that because you got like, if you're using synthetic DNA, you're using non-modified DNA and it's getting chewed up by nucleases and stuff?
It's possible. So we do recircularize. So it's not the exonucleases. I mean, Sam Kilcher has done this. We've actually done this in Pseudomonas where we make the long PCR pieces, we repackage it. But then when we move out to different organisms, sometimes like Burkholderia is very hard to transform with anything, any DNA. So it's become very difficult. And then when we move into these soil systems, sometimes you can't even isolate that bacteria. So using that bacteria specifically for rebooting is going to become a challenge. So we are actually looking actively at various paths, some of which have been mentioned here and others which we're kind of developing right now to reboot anything and to make this platform universal. We don't want to say, okay, well, I need this Burkholderia strain to redo this rebooting. I want to say, I'm going to put it in this and I'm going to get a phage out no matter what I put in. Okay, let's chat at some point.
There's a question for Gary Trouble here. Hi, Kitty. Did you try using a different induction agent? I find with soil phages that mitomycin C does not work that well.
So right now we've done mitomycin C. We have explored other ones. We haven't done it in the large scale with all of the Burkholderia. We are also working on a few other soil bacteria, and we've tried a couple others that have been published in the literature, but I'm not showing that data today. So I do agree with him that in soil bacteria, mitomycin's 50-50.
Okay, thank you so much. And we have Ivan Lyachko from Phage Genomics. Sorry.
Okay, congratulations. You guys made it. The last talk of the day. Thank you so much for having me. My name is Ivan Lyachko. I run a small company in Seattle called Phage Genomics. And what I'm going to do today is I'm going to tell you about a method that we've developed that allows us to assemble phage genomes from microbiome samples and also connect them to their hosts at the same time. So as I mentioned, we're a genomics company, so we do a lot of genome sequencing. If you're not familiar with the field of genomics, I just made a one-slide summary. So essentially what we obviously want to do is we want to sequence everything and extract as much genome information as we can from things for a variety of reasons. But this is especially difficult when you're dealing with microbial communities. And the sort of the high level summary of why is because you're taking DNA from a number of sources, a number of microbes at the same time, mixing it and sequencing it all. And now you've lost the ability to tell where these sequences came from, right? And so what you'd really like to do is you'd like to take this alphabet soup of sequence contigs and put them back in, right? So separate them out into the genomes that they come from. One of the issues, especially when you're dealing with phages or other mobile elements like plasmids, is that, if one of these organisms has a plasmid, or like there's this plasmid or a phage, even if you assemble the entirety of this genome down here, nothing will tell you that this phage came from the green cell, right? Or the blue cell or whatever, right? So mobile elements, because they're disconnected from the genome, tracing them back to where they came from is extremely difficult. And so, what we've been trying to develop and what we've developed is a method that allows you to basically separate and bin metagenomes without relying on indirect methods. So actually know which sequences belong where. It allows us to reconstruct novel genomes from prokaryotes, eukaryotes, and phages. It would be nice if we could connect mobile elements with their hosts directly in those microbial communities. It'd be nice if it didn't require culturing or cell sorting. You didn't have to buy a special machine, and you didn't need to do sort of extracting high molecular weight DNA and things like that, like you do for long read assembly. And so we have a method that does most of this. And in order to explain how it works, I have to back up a little bit. So because this is a method that was originally developed for something else. So what, when you think of your DNA, chromosomes, genomes, you think of, this is sort of the picture that comes up, but actually your genome, it looks like this, right? Genomes are packaged into these three-dimensional structures, these hairballs inside of cells. And because there's a lot of biology that goes on inside of these structures, methods have been developed to reconstruct the 3D architecture of genomes and how they fold. And what we're going to do is we're going to take one of these methods, the most common one called Hi-C, and we're going to repurpose it for something really neat. So the way the method works is you take cells when they're still intact, right? They're in their sort of three-dimensional shape, and you treat them with a cross-linker, usually formaldehyde. Formaldehyde goes in to the cells and it cross-links proteins, DNA, RNA together. And in doing so, if there are DNA sequences that are physically close together, they get cross-linked, right? You can then fragment this cross-linked chromatin, and then you do what's called proximity ligation, which is like if you've ever tried cloning a plasmid, the plasmid tries to close in on itself because the two ends are close to each other. That's what we're doing here. And so sequences will ligate that are physically close, and they create this chimeric junction between this red and this blue sequence. Okay, and then you can, there's lots more steps that I'm not showing, but basically you can extract these junctions and sequence them, typically by Illumina. And when you sequence one of these chimeric junctions, it tells you that whatever this red sequence is on the left, whatever this blue sequence is on the right, they were physically next to each other in three-dimensional space. Okay, because you're doing this millions and millions of times at the same time with Illumina sequencing, you're effectively counting how often every locus was physically close to every other locus, right? So you're saying sequence A and B were always next to each other, sequence B and C were never next to each other, and you have that for every combination of coordinates. Then you can extrapolate these into matrices, and from that information, you can, that's how you get your three-dimensional structures of genomes, okay? But now, so why am I bringing this up in this context? Because this is what the method was originally designed for, to do this 3D modeling of genomes. But it actually gives you information that is, I think, far more valuable for, at least for this community. And that is the very simple fact that because the cross-linking happens before you broke the cells, all these little junctions are happening inside the cells, okay? So when you sequence one of these red-blue pieces on the right, anytime, like whatever those blue, like anytime a red and a blue interact, you know that they must have started out inside the same cell, right? Because the crosslinks happened before you broke the cell. And that's like the most important slide in this whole deck, okay? So the idea is anything that interacts by this assay must have started out inside the same cell. And so if you have a microbial community like this and you shotgun sequence it, right, you're gonna like, you have this alphabet soup of contigs. But if you have this Hi-C proximity ligation data, you will get junctions inside chromosomes and between chromosomes as well, right? But not between cells. And so you're going to have intra as well as inter-chromosomal physical connections between DNA. And so that when you see, when you have this data, you're able to separate your metagenome because you know all these were touching each other, they belong to one organism. All these were touching each other, they belong to a different organism. Okay. And so what we've done is, you know, and this is a picture which is called the soccer ball of science. This is from a picture from our very first proof of concept that we've ever did. We published this a few years ago, but basically we took a dozen yeasts. So these are all fungal, all multi-chromosomal organisms. We mixed them together and we shotgun sequence and did Hi-C. And basically what this chart is showing, all the dots, all the nodes in this chart are contigs from the metagenome, and the edges, the lines that are connecting them all, these are the Hi-C signal. And so if you didn't do Hi-C on this, it would just, if you just did shotgun, you would just have a gray field of disconnected dots. Okay. And what you can see is that the Hi-C, the proximity ligation data, grabs them and separates them into these tight clusters, right? And we say, okay, well, what are these clusters really? And it, you know, they are the 12 organisms that we put in. So we showed even early on that this was a very accurate way of being able to take a mixed community and separating all the constituents. And remember, these are like, some of these bugs have 16 chromosomes, there's plasmids in there, there's all sorts of mobile elements, okay? And so we commercialized this. This is sort of one of the core technologies of Phage. We call it Proximeta, proximity guided metagenome assembly. And it works just like how I showed you. You take a sample, cross-link it, generate Hi-C libraries, and then from the data, do all sorts of cool metagenome things. So the first thing to point out about this tech is that because it's using direct physical information as to which sequences are contacting each other, it is inherently more powerful than normal binning. And so, so this is just showing, like, for a given sample that the method essentially is designed to, it will always outperform traditional binning tools because it uses this extra bit of information about what's physically touching. The nice thing is it works on all sorts of things that normal binning doesn't work on. Algae, archaea, protists, like it doesn't care. It's just grabbing sequences that are touching each other and putting them into mags. And so we're able to basically pull out all sorts of genomes that you just can't do in normal binning experiments. And here's an example. This is our platform spits out these reports. This is from a fecal sample, a human fecal sample. The way you read this is on the x-axis, every tick mark, every point is one of the genomes, one of the mags that came out of the sample. The blue dots represent completeness of these mags according to a software called CheckM. And so you can see in this case, we have about 60 genomes that are 95% complete or more, according to CheckM. From, again, one sample, there's no dereplication. We're not doing any database stuff, right? Contamination is orange and it's low. The other thing is that all of these gray bars, they're a measure of novelty. So are these things, we use RefSeq just to be on the safe side, but so anything that's a gray bar is basically something that's not in RefSeq. Okay, so what's nice is this technology is able to get you genomes, whether they're old, whether they're new. It pulls out a lot of these things in the tail that are local. They're actually not, they're like, you can tell they're complete genomes. They're for things that CheckM has not been trained on because there's some weird like archaea or something like that. And so CheckM just gives them terrible quality scores but they're actually pretty good. Now you say, okay, well, but I'm a fancy scientist. I have long read data, right? I'm going to do PacBio or Nanopore. This is what happens when you, we just published this. This is what happens when you combine this with long read assemblies. So this was a PacBio hi-fi assembly of a sheep poop sample with, plus our Hi-C stuff. And so what you can see is there are 400 mags, 400 genomes are coming out of the sample that are more than 90% complete. There are also all sorts of other mobile elements in here and stuff like that. And the other cool thing that I like is that notice that almost every point has one of these gray bars, right? So in the human sample, you get, it's been studied to death. So you have lots of organisms that have already been cultured. But as soon as you leave the human space, you get like everything is novel. Like all these bugs are not in RefSeq. So it's a really good way of really digging into that dark matter, quote unquote, of the microbiome. But we can actually go further. So remember, I mentioned mobile elements and how difficult they are to track in microbiome samples because they jump from an organism to another one and they're not necessarily part of the core genome. Well, let's take a sample. This is something we published a couple of years ago. This is a wastewater sample that has tons and tons of microbes in it. But what we can do is beyond just looking at the genomes, we can annotate antibiotic resistance genes, plasmids, and see who they're touching, see who they're connecting with, right? And then that data, this is a shot from that paper. Basically, this is just the proteobacterial mags that we pulled out of the sample. They're arranged in this tree in the middle. And these concentric rings on the outside, these are plasmids, integrons, and antibiotic resistance genes. So if you have a tick mark, that means that this antibiotic resistance gene was present inside of this host, okay? And if you have them in a row like this, that's telling you that this AMR gene is present in all these different hosts, right? What's cool is this is not limiting them to one per one. It's not like this sequence belongs to this organism. An AMR gene or a plasmid can be present, or a phage can be present in many hosts. And then you can also look down this way. If you have a stripe going this way, that means that whatever this bacteria is, whatever this microbe is, it has all these different plasmids in it. So you can detect multi-drug resistance, et cetera. But what about phages, right? So you can actually do the same thing with phages. This was originally demonstrated by Ramon Cazul's lab in Paris. So they've shown that it's analogous. So here on this horizontal axis, these are phage genomes and contigs, and these are hosts, bacterial hosts. This was from